Hello everyone, my name is Emma Perry and today I'm going to be reviewing the article Mechanism of Inactivation of Enveloped Viruses by the Biosurfactant Surfactin from Bacillus. To start off with some introduction on the background a little bit, biosurfactants are amphiphilic compounds that are produced in living bacteria which accumulate between fluid phases. So this allows them to reduce surface and interfacial tension. This article is looking for a better and safer treatment towards human viruses through the use of biosurfactants. So the study works with the bacterium Bacillus subtilis, so B sub. It's a soil bacterium that synthesizes biosurfactin. And the surfactin is a cyclic leptopeptide antibiotic which is made of several fatty acid chains. So this surfactant possesses an amphiphilic structure, which makes it one of the strongest biosurfactants. You can see there's a figure which shows its structure. So the hypothesis and goal of the research is to study the effect that surfactant has on the development and survival of various viruses. It's also through the experiment, they're going to observe temperature and concentration dependent inactivation on sued herpes, a model which they're using for human herpes. And their action plan is to create an antiviral treatment with the surfactant for certain human viruses. Using biosurfactants this way is something that's very big now, and it is also very large in the agriculture industry, working on getting rid of viruses that attack crops and plants. Materials and methods. So this study is going to be utilizing several viruses which they will have the surfactant up against. So those are herpes type 1 and 2, vesicular stomatis virus, samian immunodeficiency virus, Semliki forest virus, feline calic virus, and murine encephalomycarditis virus. And here is a picture of herpes cells under a microscope. So the study is going to observe the concentration-dependent virus inactivation by infecting each virus with varying amounts of surfactant. They will also observe temperature-dependent virus inactivation by taking one of the viruses and 80 microliters of surfactant and exposing them to temperatures ranging between 5 degrees and 30 degrees Celsius. The study is going to observe the results of the disintegration of viral envelopes. They're using electron microscopy, titration, and cytotoxicity assays to determine all of the results that they find. Moving into the results, so the overall effect of surfactant on cell proliferation, they found that surfactant inhibited cell growth for all concentrations higher than 20 microliters of surfactant. And they found that proliferation was inhibited by 50% of concentrations and completely inhibited at all concentrations that were higher than 70 microliters. So when observed if the surfactant would have a stronger inhibition on cell infection or virus replication, it was found that surfactant has a strong effect on the virus particles alone before they have infected the cell. So this would be the free cell virus within the body, not inside one of our cells yet. So figures 3 and 4 show both the temperature and concentration, and you can see that concentration of overall temperature increased inactivation of the viruses. So figure 3 is on the top, shows linear regression for inactivation of sued herpes when observing concentration dependent of the surfactant. So the higher the concentration, the higher the inactivation of the virus. Figure 4 is underneath figure 3, and it shows the inactivation of sued herpes virus biosurfactant at temperature. So as you can see, the higher the temperature, the higher the rate of inactivation. Then the article shows us what disintegration of the viral envelope looks like through electron microscopy. We can see here in A and B, you can see that the envelope, the arrows, there's black arrows in figure B indicating to where the envelopes have been broken. This is following incubation of surfactant and sued herpes virus. And you can see that the lipid membrane of the virones was detected. So those are complete infected form of the virus outside the host cell with a core of RNA or DNA and a capsid. So these are just the virus cells alone in the body. So moving on to the discussion, the research was able to verify that in a control study, surfactant can inactivate envelope RNA or DNA viruses such as herpes and retroviruses. However, they highlighted that a virus family such as herpes viridae might contain components to its envelope that specifically stimulate surfactant to disintegrate its membrane. So this statement may be very viable because surfactant failed to inactivate SFV, which was Semliki forest virus, that they used as a model for Hep C throughout the study. So it might be true that components of that envelope were able to stimulate the surfactant in herpes, but it is not in other viruses. So again, looking at the breakdown of the viral envelope, they were able to see that the lipid membrane was disintegrated, causing the free virus to be inactivated. However, the microscopy was able to show that when surfactant is inoculated with cultures at any point in replication of the virus, there was no harm done. So this basically means that surfactant is only able to inactivate cell-free viruses before absorption or penetration, but not at all during their cell replication cycle. So if this were to be used to fight off a virus, any viruses that are in the stage of replication within the body will not be affected. Only cell-free viruses. So to talk about the cellular mechanism that this paper is incorporating, we can look into previous studies. It was observed that surfactant inserts into the outer layer of the lipid membrane bilayer, causing permeability changes. So these changes may be the formation of a high concentration of ion channels that disrupt the lipid membrane system. This could be due to the high amount of fatty acid chains within the biosurfactant, and in this case the biosurfactin, because it was a very strong structure, monoglycerides as well, which disintegrate the envelope and act as antiviral agents. So the biosurfactants fatty acid chains are what is breaking down this lipid membrane, so that cellular mechanism is what this study is observing. So overall, in conclusion, inactivation success rate of surfactant is dependent on its environment. This refers to the reaction that is happening in the medium as the virus decreases, the lipid and protein content increases. So as the surfactant is breaking down the lipid membranes of viruses, as the environment changes, what the surfactant does is also going to change. So though the surfactant isn't completely safe to use for direct human viruses yet, it is a very helpful tool in the lab. This is because of increased proliferation rate that surfactant has on mammalian cells. So it allows for laboratories to have quicker results within their research because everything happens a lot faster. So overall, there is a lot more research that is yet to come in this field. As I mentioned earlier, this is very big in the agriculture industry right now. finding biosurfactants within bacteria that live in the soil, around the biosphere, and plant roots. It's been a large help, and it is also commercially sold in some circumstances. So it's a very large field, and it's very interesting to study. So I hope you enjoyed, and here are my references, and thank you very much.